For those who don't know me, which is probably most of you, my name is Benjamin Richards. I'm a research fish biologist with the Stock Assessment Program in the Fisheries Research and Monitoring Division at the NOAA Fisheries Pacific Islands Fisheries Science Center in Honolulu. And what I'm going to be talking about today is a little bit about the fishery-independent surveys that we run and how we are starting to incorporate machine learning and computer vision in our stock assessment process. Okay, so a little bit of an overview of the talk. So I'm going to start out talking about the bottomfish fishery-independent survey that we run here in Hawaii, a little bit about the data needs that we have for stock assessment, the survey gears. In the interest of time, I'm not going to go into the details of stratification, but I am going to talk about the abundance and biomass estimates that we produce, and then how we're using machine learning to help with some of that heavy lift on the data side. And I'll detail two main products that have come out of the NOAA Fisheries Strategic Initiative on automated image analysis, one being called CoralNet and the other being called Biomass. Any questions before I get started? Okay, hearing none, just a little bit of a background for those who aren't familiar with stock assessment. The stock assessment process is the process by which NOAA Fisheries produces scientific information on the status of commercial fisheries. That stock assessment uses three main data types: information on fisheries abundance, which is basically how many fish in a particular fishery are there out there in the ocean swimming around, the biology, how fast are they growing, how long do they live, how many offspring do they produce, and then catch, how many fish are being removed from the ocean every year. Those are what are known as the ABCs of stock assessment. That information flows into a population model, which is used to generate an estimate of stock status, which is then provided to the management agencies to make decisions on annual catch limits. So everything that we do governs, in essence, how many fish commercial fishermen are able to catch each year for any particular fishery, which then dictates how many are available for you at the grocery store. So what I'm going to be talking about mainly today is some more information on how we get at this abundance index. How many fish are there out there swimming around in the natural environment? And the principal sampling tasks of a fisheries survey are basically to get counts by species, so how many fish of any particular species are there out there in the water, and then the individual lengths for each fish. And we need the length information because we have formulae that translate the length of any particular species to the weight of that species. And since most commercial fisheries report their catch in terms of metric tons or pounds, everything that we do in terms of generating abundance estimates is generally done in terms of generating weight estimates and abundance in terms of what's called biomass. So just a quick example of a fishery-independent survey. As I said, we run fishery-independent surveys for a large number of commercial stocks, both in Hawaii, Alaska, New England, Gulf of Mexico, California coast. So there are 6 NOAA Fisheries Science Centers, the Alaska and Northwest Fisheries Science Center in Seattle, the Southwest Fisheries Science Center in San Diego, the Pacific Islands Fisheries Science Center here in Hawaii, the Southeast Fisheries Science Center in Miami, and the Northeast Fisheries Science Center in Woods Hole, Massachusetts. And pretty much all of the science centers run fishery-independent surveys of one form or another for the stocks in their jurisdiction. For Hawaii, I'm going to be talking about the bottomfish survey, or the bottomfish stock, which is 6 species of deepwater snapper and 1 deepwater grouper that form the main commercial fishery for Hawaii. And the area that those fish inhabit is depicted by this gray polygon that surrounds the main 8 Hawaiian islands. That references a depth range of about 75 to 400 meters. It covers a total area of about 6,500 square kilometers, stretching 550 kilometers from the island of Kauai in the northwest to the big island of Hawaii in the southeast. We divide that area into a survey grid of about 500 by 500 meters. So there are about 26,000 primary sampling units within that domain. We use a variety of survey gears to collect data on the fish stocks. The first two here on the left, research fishing and drop cameras, are the two that I'm going to be talking about today, and primarily the drop cameras. So we use these two gear types. We have commercial fishermen that go out and fish for us using standard hook and line gear. They capture the fish, bring them on deck of the vessel, and then they're easily able to identify the species, measure them, and count them. Then the drop cameras, we have a stereo video camera system called the BotCam, or the MOUSE, which is the Modular Optical Underwater Survey System. It's a low light, highly sensitive ambient light camera. It goes down to a depth of about 300 meters. And at each survey location sits on the seafloor for 15 minutes recording stereo video. When that camera, after the 15 minutes, that camera is recovered to the surface, redeployed at another survey location. We have 10 of those cameras that are basically leapfrogged through the survey domain by several vessels over about a 3-week period. This is what a typical survey might look like. This was actually the fall 2018 bottom fish survey. So the red dots represent about 450 fishing locations. The green dots represent about 80 camera locations. And then the yellow dots are about 15 locations, 12 to 15 locations where both gears were deployed. So as you can see, there's quite an extensive coverage. We do get quite a lot of data back from the surveys. When the fishing data and the camera data is processed, we're able to create abundance and biomass metrics, basically taking the mean number, the mean biomass of each species per survey stratum, applying a weighting factor, and then getting a per sample average catch per unit effort and a mean biomass per sample across the survey domain. And that's the information that is passed into the stock assessment to help give a stock status reference for managers. So that's just a little bit of a background of the type of data that we're trying to collect and how it's used. As I mentioned, one of the primary survey tools is the MOUSE, the Modular Optical Underwater Survey System. It's a stereo camera system. So as you see here, there's one main base bar that serves as the main structural component of the camera. On the left and the right, there's two video camera heads. Right now, they are Allied Vision low light cameras, so we don't use any artificial light, and we can record down to about 250 meters in Hawaiian waters. There's an onboard digital video recorder and a battery to make it a completely autonomous system. This is what it looks like sitting in the water. There's an anchor that goes down to the bottom. The system sits about 3 meters off of the seafloor, has some subsurface floats that keep it oriented vertically in the water column so that we can get a good video picture of what's going on in the environment. Give you an example of what some of that looks like. Here's some footage of various different fish species here in Hawaii. As you can see from the camera footage, we get some really nice pictures. And because the camera is a stereo camera system, not only can we identify and count each of the species, but in software we can put a dot on the nose and a dot on the tail of each fish in both the left and the right camera system, and use some Euclidean geometry to automatically generate the length of each individual fish, which we can then translate into a weight or a biomass metric. So optical camera technologies like this are really nice. They're fishery independent. They're non-extractive. We're not pulling any fish out of the environment, which would then be unavailable for commercial fishermen. It doesn't take a whole lot of skill to deploy the camera systems. You don't have to be a really good fisherman. There's not a lot of bias from one camera to another, as there might be from one fisherman to another. So we can reduce the measurement and calibration errors, and we can also create an auditable data stream that can be looked at and queried in the future. The main problem is that, as we all know, they create really, really large data sets. From a 3-week survey mission, we come back with about 9 million images. As I said, they sit on the seafloor for 15 minutes, recording at 24 frames a second. And it's a lot of imagery to go through. As with all of the optical technologies and camera systems that we're using, that's a pretty common problem. So starting in 2014, NOAA Fisheries initiated the Strategic Initiative on Automated Image Analysis with a mission to develop guidelines, set priorities, and fund projects to develop broad-scale, standardized, and efficient automated analysis of still and video imagery for use in stock assessment. That panel was a 12-person panel chaired by myself, one representative from each of the 6 fisheries science centers, and then 6 representatives from private industry and academia, spanning Monterey Bay Aquarium Research Institute, Kitware, Inc., which is one defense contractor, UC San Diego and Dropbox, Stanford Research Institute, Los Alamos National Labs, University of Victoria, and Google. And so our task, our goal, was to create a computer vision toolkit. It's basically a software tool, application, or set of algorithms that was easily usable by non-computer scientists, people who were not familiar with command line, basically wanted to count fish and produce some numbers, but weren't coders in and of themselves. That effort ran from 2014 until 2018, and we developed 2 main software products out of that effort. One is called CoralNet, which is a web-based tool for the classification of benthic habitat photos, so primarily coral reefs. So if you see here on the left and the right is what's known as a standard photo quadrat, which is just a downward-facing digital still image of the coral reef. You can see some corals, you can see some crustose coralline algae, you can see some algae. And what CoralNet does is it automatically generates a random set of points across the imagery. So you can see that by these sort of yellow crosses with the circles around them, and then green and then purple. And what a user, a typical Benthic ecologist would do is, in the standard manual method, they would go through and classify or identify what substrate type lies underneath each of those random points: a coral, an algae, a crustose coralline algae, sand, things of that sort. And that's how it's been done for years, is just manual annotators going through and annotating each of those points. With CoralNet, we replicated the look and feel that they'd been used to using for years, but we implemented it on Amazon Web Services, and behind the scenes we have a deep learning algorithm that is ingesting all of the manual point annotations into a learning model, and basically learning how to classify each of the species or taxonomic categories that the analyst has developed. Over a period of time, as the model continues to improve, the analyst will start to see the top 5 suggestions that the model has for each of the points, along with the confidence in that classification. So the analyst can choose from those suggestions or provide an alternative suggestion. That iterative query and refinement goes back into the model, and the model starts to generate its own accuracy curve. When the reported accuracy reaches a level that is acceptable to the human analyst, they can then turn the model loose on the rest of their imagery and then just spot check any annotations for which the model has a low confidence. Last year, we published a paper looking at the pairwise differences between human analysts and the CoralNet algorithm, and we found that for classifications at the functional group level, so that would be coral, algae, sand, things like that, we're at a point where we don't really need human interaction in the data analysis at this point. There's no significant difference between the CoralNet model and the human analyst model at the functional group level. At the species level, the model's still not as good. Partially that's because a lot of the human analysts aren't necessarily that good at speciation, so the training data is not as clean as we might like it, but it's continuing to improve. The next piece of software that we developed is called Biomass, and Biomass is an open source software toolkit that's currently sitting on GitHub that provides a series of tools for the ingestion of a very broad range of image types, primarily targeted to in situ imagery or videos of fish in the natural environment, similar to what I showed for the mouse camera system, but is quite robust and able to ingest a wide variety of images from fish in the water, eels on ice, penguins in their rookeries, all sorts of things like that. All based on deep learning. There's capabilities for object detection. There's a tool for generating training data through IQR or manual annotation, object tracking. So if you have images that have time associations from one to another, such that they're in a sequence, unique targets can be tracked from frame to frame, so bringing motion into the classification algorithm. Automated detections over time, so this is an example after training of some of the abilities to automatically detect and track individuals within a video frame. So again, this is no human interrogation at all. So it's just showing some of the automated detections and segmentation, tracking of individuals over time throughout successive frames. And what that allows us to do is to automatically generate a detection over time curve. So if you can imagine in a manual annotation method, which is what we've been doing to date, we have a bank of human Turkers, mainly graduate students and undergraduate students, that watch each of these 15-minute videos, and they generate an abundance metric using what's called the max-n method. So they watch the whole 15 minutes. They find the video frame that has the highest number of fish for any particular species, and then they enumerate that frame. So in this example here, what you see is time on the x-axis running from 0 to 15 minutes, and then the fish count, or the number of detections, on the y-axis. And so you see here the maximum number of detections, which was 12, occurred at about minute 11 in that video. And this curve is automatically generated with no human interaction at this point. So rather than watching the whole 15-minute video, they could just say that this count was a count of 12 at this particular survey site. If they wanted to interrogate the video themselves, rather than watching all the way up to minute 11, they could just immediately scroll to minute 11, double check that count, and then move on to the next video. Also particularly useful for what we call sorting the wheat from the chaff. A lot of the videos that we return with don't have any fish at all, no detections. So this is a very quick and easy way of separating the videos that have detections from those that don't have detections for allowing our human analysts to triage the video and make the most use of their time. We also have some measurement capabilities. This is an example from a pollock survey up in Alaska. So here they have a stereo camera system that's mounted sideways in a traditional trawl net. Here you're seeing both the left and the right frame from a stereo camera system. The software is able to match detections from the left and the right frame, and then use geometry to calculate the length of each individual fish from the longest axes from each of the detection boxes for each individual. So the current status, as I mentioned, both CoralNet and Biomass are both in the wild. CoralNet is running on Amazon Web Services and accessible through coralnet.ucsd.edu. Currently, there's about 760,000 images that have been uploaded and have been annotated, comprising about 27 million, 28 million annotations. Biomass is also in the wild on GitHub. There is both the source code and downloadable binaries with user guide and manuals. We also just published earlier this year a NOAA technical memorandum, so that's NMFS 83, that outlines basically our efforts from 2014 to the present and gives examples of Biomass and CoralNet usage and other data streams coming out of each of the 6 fisheries science centers. Recently, as I'm sure everyone on the phone is aware, there's been increased effort and increased attention following the executive order on maintaining American leadership in AI. NOAA has set up an artificial intelligence executive committee with a strategy and implementation plan. There's also a NOAA Fisheries AI and machine learning working group that's being stood up with a plan for a workshop on machine learning and computer vision in the second quarter of 2020 in Silver